Hello everyone. In this video, we are going to discuss the Common PG Entrance Test 2022 for the subject Biotechnology. Okay, and the subject code of Biotechnology is 48. Okay, so let's start from question number 1. Okay, so the life cycle of marine alga sargassum is. So the life cycle of marine alga sargassum, it's a kind of a diplontic. So option A will be the correct one. Coming to question number 2, which of the following is correct order of evolutionary history of human? So in case of human, so Peking man, then Heidelberg man, then Neanderthal man and Cro-Magnon man. So this is the correct order of the evolutionary history of human. So option C will be the correct one. Coming to question number 3. How many molecules of NADPH and ATP are required for the fixation of 6CO2 molecule to generate one molecule of glucose, that is 6S-X12O6. So here we need 12 NADPH and 18 ATP. So option B will be the correct one. Coming to question number 4. A placid plant cell with 0.3 molar solute concentration, here psi S is equal to minus 0.732 megapascal, placed in a beaker containing zero point 0.1 molar sucrose solution. Here the solute psi S is the minus 0.244 megapascal and the placid cell became turgid. Okay, so with the increment of the cell size by 25%. So what is the hydrostatic pressure, that is psi P exerted on the cell? So here after calculation we get 0.342 megapascal. So option B will be the correct one. Coming to question number 5. Which one of the following is considered as missing link between reptiles and birds? So it is the Archaeopteryx is the missing link between the reptiles and birds. So option A will be the correct one. Next, coming to question number 6. The process of formation of seeds without fertilization in flowering plants. It is very easy question. So that will be your apomixis. Apomixis is the formation of your seeds without fertilization. So that will be option B will be the correct one. Coming to question number 7. The hierarchical organization of taxonomic status of an organism based on evolutionary lineage is known as your phylogenetics. So option D will be the correct one. Coming to question number 8. The genes that are related by vertical descent from common ancestor and encode proteins with the same function in different species is known as orthologous. So option A will be the correct one. Coming to question number 9. The hormones of adrenal medulla are the modified amino acids. So option B will be the correct one. Coming to question number 10. Okay. So during gastrulation, the developing embryo undergoes recognition of the cells into a multi-layered structure, and each of the layer will differentiate it into distinct parts in the fully developed organism in due course of time. Which one of the following layer is responsible for the development of skin and nervous system? Okay, so it's a very lengthy question, but question is only answer is present only in this last sentence. Okay, so all the before sentence one, there are multiple sentences are there. Okay, so we can ignore this kind of your multiple sentence. Okay, so this multiple, due to multiple sentence, it's a kind of a lengthy, and that makes the student in confusion. Okay, so here Which one of the following layer is responsible for the development of skin and nervous system? Okay, so for skin and nervous system, ectoderms are responsible. Okay, so option C will be the correct one. Coming to question number 11. Which one of the following condition make a biological process spontaneous across all temperature? Okay, so in order to the biological process to be spontaneous, the process is enthalpically opposed by entropically favored. That should be entropically favored. So option C will be the correct one. Coming to question number 12. Dex is the most abundant element in the human body without any infection and deformities. So that will be your carbon. So option D will be the correct one. Coming to question number 13. Which one of the following bonds is present in both DNA as well as protein? So as we know, the glycosidic bond is present between your in between your base and your sugar, okay, and peptide bond present between your two amino acids, okay, that is not present in DNA. Phosphodiester bond present in only DNA, okay, but the hydrogen bond is present both in DNA and protein. So option D will be the correct one. So in between your base pairs AT and GC, hydrogen bonds are there. Okay, in between amino acids. Okay, and in the amino acids you will get hydrogen bond. So option D will be the correct one. Coming to question number 14. Tryptophan is an aromatic amino acid which synthesized in plants by Cpet or Cpimic acid pathway. The sequence of its synthesis from phosphoenol pyruvate PEP and Crystose 4 phosphate, okay, E4P. Each the correct sequence will be your PEP plus E4P, that will be your erythroch 4 phosphate, DA, DHP, Cykimic acid or Cykimate, then corismate, anthralinate, and tryptophan. So option A will be the correct one. Coming to question number 15. So how many adenine base are present in bDNA fragment of 100 base pairs? Okay, with 60% GC. So here the GC content is 60%. Okay, so first we need to know the amount of or number of bases. So here 1000 base is there. So 1000 base means that will be 2 1000 base pair means that will be 2000 base pair. So out of 2000, so 60% will be GC. So uh, out of 2000, that will be from 2000, 60% will be your 1200. Okay, so out of that, uh, 800 will be A plus T. So definitely A equals to T. So by we need to divide it by 2. So that answer will be 400. So 400, that will be 4 into 10 to the power 2 will be the correct one. Coming to question number 16. The beta seeds are example of the secondary structure. These are the beta seeds are the secondary structure. So option B will be the correct one. So coming to question number 17, oxidation of palmitic acid, that will be your carbon 16 carbon compound, requires dex rounds of beta oxidation and yields dex number of acetyl coenzyme A. So that will require 7 round of beta oxidation and that will eat 8 number of acetyl coenzyme A. So option A will be the correct one. Coming to question number 18. In Michaelis-Menten equation, the enzyme kinetic curve is always hyperbola. So option C will be the correct one. Coming to question number 19. A phage capable of destroying host cell is known as your virulent phage. So option A will be the correct one. Coming to question number 20. Okay, coming to question number 20. The hospital acquired infections are known as your nosocomial infection. So option C will be the correct one. Coming to question number 21. Which one of the following is not an antibiotic? So amoxicillin antibiotic, cephalosporin antibiotic. Okay. Clarithromycin is also antibiotic. Okay. So option D will be the correct one. Coming to question number 22. Coming to question number 22. Genetic recombination in E. coli occurs through all of the above: transformation, conjugation, transduction. Question number 23. So gram staining differentiates Staphylococcus aureus, that is gram positive, and Escherichia coli, that is gram negative, due to presence of multiple inner connected layer of peptidoglycan without outer membrane in Staphylococcus aureus. So option A will be the correct one. Coming to question number 24. The phase of bacterial growth curve where exponential multiplication of E. coli cells occur is known as log phase. So option B will be the correct one. Coming to question number 25. The industrial production of vinegar through fermentation involves the yeast with acetic acid producing bacteria. So option C will be the correct one. Coming to question number 26. The antibacterial action of penicillin is due to its effect on cell wall synthesis. So option B will be the correct one. Coming to question number 27. The major components chromosome in DNA and protein. Among them, DNA is considered as genetic material because DNA possesses all of the above: autocatalytic property, heterocatalytic property, mutability. So option D will be the correct one. Coming to question number 28. Which one of the following is used as template strand during transcription of E. coli? So that will be your antisense strand. So template strand is also known as antisense strand. Okay. Coming to question number 29. Which one of the following is not a mechanism of post-translational modification? So in the post-translational modification, so proteolytic cleavage is there, apurinic cleavage is there, O-linked glycosylation is there, but not intine splicing is there. So option A will be the correct one. Coming to question number 30. The interaction of triplet codon of mRNA with the anticodon of tRNA is stabilized by your hydrogen bond. Coming to question number 31. Which one of the following enzyme is required for the photoreactivation of TT dimer, thymine dimer? Okay, so that will be your DNA photolyase. So if you give photolyase in presence of blue light, so TT dimer can be removed. So option B will be the correct one. Coming to question number 30. Termination of DNA replication in E. coli is triggered by. So that will be your TOS protein. Coming to question number 33. Tryptophan operon in E. coli is regulated by unique phenomenon of, that will be repression. Okay. So lac operon is the inducible operon and tryptophan is the repressible operon. So in that case you can say the tryptophan operon, it's a kind of a repression. So coming to question number 34. The cDNA, that will be complementary DNA clone, differs from genomic DNA or gDNA by absence of intron. So option C will be the correct one. Coming to question number 35. Resolving power of microscope can be increased by using an illumination of shorter wavelength and increasing the numerical aperture. So option B will be the correct one. Coming to question number 36. The fluidity of the plasma membrane increases with increase in unsaturated fatty acids in the membrane. So option B will be the correct one. Coming to question number 37. Which of the following plasma membrane receptors activate signaling pathways usually by forming molecular dimers that result in protein phosphorylation reaction upon binding of their specific ligand? So option C, that is receptor tyrosine kinase, will be correct one. Coming to question number 38. Which one of the following cell organelles is semi-autonomous in all eukaryotes? So, semi-autonomous organelle, that will be mitochondria in all eukaryotes like your plants and animal. chloroplast is also say kind of your semi-autonomous organelle, but that is in case of plant. So option B will be the correct one. Coming to question number 39. Which of the following cytoskeleton proteins have an almost all helical secondary structure? So that will be keratin. Option C will be the correct one. Coming to question number 40. Assuming Hardy-Weinberg equilibrium, the genotype frequency of heterozygotes. So genotype frequency of heterozygote will be, okay, here the allelic frequency of 2 is given, P will be 0.6 and Q is the 0.4. So for the heterozygotes, that will be formula will be 2 PQ. If you multiply 2 into 0.6 into 0.4, so that will be 0.48. So option B will be the correct one. If there will be a question asks about your homozygous, okay, homozygote, so that will be your P square or Q square. Coming to question number 41. Mitochondrial DNA, that is mtDNA, is advantageous for evolutionary studies because mitochondrial DNA is inherited maternally, which paves the way to elucidate phylogenetic relationship. So option A will be the correct one. Coming to question number 42. If one gene marks the effect of other gene during dihybrid experiment, then the mode of gene action or gene interaction is considered as the epistatic gene action. So option D will be the correct one. Coming to question number 43, a cross between two true breeding lines, a plant, one with dark blue flower and one with bright white flower, produces F1 offspring that are light blue. When F1 progeny are selfed, the F2 progeny have segregated in the ratio 1 is to 2 is to 1. That will be 1 dark blue, 2 light blue and 1 white for its flower color. So what genetic phenomenon is considered with this result? So with this result we can say it's a kind of a incomplete dominance. So option C will be the correct one. Coming to question number 44. Place the following 5 steps of DNA isolation and purification in correct order. So for the process of your DNA isolation and purification, first we need to go for breaking of the cell tissue, okay, under suitable cell lysis buffer. So this will be first. Then we need to go for the purification of DNA and RNA for and proteinase K. Okay, so this will be second one. Third one will be separation of DNA by phenol, chloroform, isoamyl alcohol, that will be 25 is to 24 is to 1 extraction. So that will be third. Okay, then ethanol precipitation of DNA and solubilization of DNA with TE buffer. This one 4. And assessment of the quality of by DNA by your UV spectrometry, photometry. This will be next. Sorry. Okay. So option D will be the correct one. Coming to question number 45. Antibodies are. So antibodies are glycoproteins. So option C will be the correct one. Coming to question number 46. Light chain and heavy chain of antibodies are joined by your disulfide linkage or disulfide bond. So option D will be the correct one. Coming to question number 47. So DEX gives a primary immune response in human being. So that will be first immune response or immunoglobulin that will be produced during the process of your infection or any kind of foreign particle. So that will be your IgM. So option B will be the correct one. Coming to question number 48, the monoclonal antibodies recognizes the epitope. Okay, so here there is a two term is there. You need to remember, one is your epitope, another one is the paratope. Okay, so basically what happened? So epitope that will be present in the antigen, and that is the antibody binding site, and paratope present in the antigen, and that will be your antibody. Paratope present in the antibody, and that will be your antigen binding site. Coming to question number 49, which one of the following is the component of the aeration and agitation system of a fermenter? That will be impeller, baffles, spagers. Okay, so option D will be the correct one. Coming to question number 50, which one of the following cloning vector incorporated highest length DNA fragment? So that will be your cosmid. So option D will be the correct one. Coming to question number 51. So DNA ligase is. So DNA ligase is an enzyme that joins fragments during cloning of gene. So option A will be the correct one. Coming to question number 52. DEX used as proofs during DNA fingerprinting of human being. So that will be your SSR sequence. Coming to question number 53. Which of the following is used as selection marker for the plant cells transformed with an agrobacterium tumefaciens based vector? So that will be your neomycin phosphotransferase 2, NPT2. So option A will be the correct one. Coming to question number 54. Which of the following is a direct gene transfer method used in plant system? So that will be your particle bombardment or gene gun method. So option B will be the correct one. Coming to question number 55. A non-anchorage dependent cell is host to Baltimore type 5 virus. If the cells are grown in the presence of your carbon 14 level deoxyuridine diphosphate, that will be C14D-UTP. Which of the following statements are correct? So here none of the statements are correct. So option D will be the correct one. Coming to question number 56, which one of the following statement is true for enzyme? So as we know, the holo enzymes, that is the active one, okay, after binding of your cofactor with the inactive FO enzyme, that will make the holo enzyme. So option A will be the correct one. Coming to question number 57, which techniques were used for quantitative estimation of DNA in plant extract? So that will be your UV spectrophotometry. So that will be option C will be the correct one. Coming to question number 58, which one of the following methods used for exponential synthesis of targeted DNA fragments in vitro? So that will be your polymerase chain reaction, or PCR will be the correct one. So option B will be the correct one. Coming to question number 59, in agar gel electrophoresis, PCR amplified DNA samples migrate toward anode from cathode. So option B will be the correct one. From cathode, it migrates to anode. So option B will be the correct one. So NCBI stands for National Center for Biotechnology Information. So option C will be the correct one. Coming to question number 61, which one of the following is not coming under intellectual property right? So patents is coming under intellectual property right. Okay. So copyrights also there, trademarks is also there, but geographical distribution is not. But that one thing is that geographical indicators, okay, are there. So option D will be the correct one. Coming to question number 62, secondary metabolites including alkaloids are produced through. Okay, so basically they are produced through the root hair cells. So root culture will be the correct one. Coming to question number 63, the first vaccine developed through animal cell culture technique was your smallpox vaccine. So option C will be the correct one. Coming to question number 64, which of the following is emerged as an international issue through the advent environmental biotechnology? So global warming and marine pollution. So option C will be the correct one. Coming to question number 65, secretory cells that release relatively large quantities of glycoproteins contain relatively large amount of ROP endoplasmic reticulum, RER. So option D will be the correct one. Coming to question number 66. So transition type of gene mutation represented by G, that will be your GC substituted by AT. Okay, so there are two kinds of your point mutation. One is your transition mutation, and another one will be transversion mutation. So in the transition mutation, so one purine will be replaced by purine, and one pyrimidine will be replaced by pyrimidine. So in case of a transversion point mutation, that will be one purine base pair will be replaced by pyrimidine, and one pyrimidine base pair will be replaced by purine. So option A will be the correct one. Coming to question number 67, which set of following gene components are transcribed in eukaryotes and are present in the mRNA but are not translated to amino acid. So that will be your introns 5 dash UTR on translated region, 3 dash UTR on translated region, and 3 dash or 3 prime poly A tail. So here it's a kind of a tricky question. So wherever you are getting exon, you need to reject it. Okay. So because exons are the coding sequence. So option C will be the correct one. Coming to question number 68. Find the range, mean, median, and mode of the following data. So here first we need to arrange in the ascending order. Okay. So here for the calculation of your range, that will be your higher value minus lower value. So if you go for the range value, that will be your highest value is 21 minus 1, that will be 20 will be the correct one. But here there is a range is there, that will be 5 to here by mistake this should be 1 to 21, and mean will be 11, median will be 10, and mode will be 21 because the frequency of 21 is high. Coming to question number 69. If all the points of a scatter diagram lie on a straight line moving from left bottom to right top corner, the correlation is called as perfect positive correlation. So option B will be the correct one. Coming to question number 70. Which one of the following genetically modified crops is based on antisense RNA technology? So there is a tomato, that is a flavor saver tomato, that is based on the antisense RNA technology, and that's a kind of your genetically modified crops, GM crops. Okay, so option C will be the correct one. So here we have completed from starting from question number 1 to 70 of your biotechnology question, Cpet entrance 2022. So there is a most important information for you guys. So there is a for the announcement, important announcement you can say. So for the year that will be 2023, for the year Cpet and Central University, Life Science, Zoology, Botany, Environmental Science, Biotech, Microbiology, okay, EVS, okay, and GATE, we are starting the new classes batch. Okay, so thank you all of you. Thanks for watching the video. Thank you. Do like and subscribe and share with your friends.